Hello and welcome to the WeActually Online Academy. My name is Chris and I'll be your tutor for today. In our previous video we looked at question 4 and in this video we'll look at question 5 of the Cambridge AS Biology paper 2-1 winter 2019 course code 9700. Question 5. Myasthenia gravis is a condition that results in muscle weakness affecting the immune response. Explain what is meant by the term immune response. Okay, so if we remember, we've got primary and secondary immune response. This is due to the effect of a foreign body or antigen or pathogen or organism that's made it into your body that shouldn't be there. And that stimulates the immune response, either the activation of the lymphocytes or production of antibodies, and that are very specific to a particular pathogen. So you could say that it's a response, okay, to what's called, a non-self, or you could say foreign antigen, okay, and this therefore activates lymphocytes. Yeah, this is going to be the chance where they'll start figuring out the specificity to target a particular antigen, and there hopefully would stimulate a secondary response if you had the secondary encounter with the pathogen. Okay. Question B. Myasthenia gravis is an autoimmune disease which disrupts the cell signaling pathway involving muscle cells. Suggest how the immune system acts to disrupt the cell signaling pathway. Okay. So what happens here is your B lymphocytes. Okay. What happens here is that they are unable to recognize your own muscle cells. Okay, so they don't recognize your own body cells' antigens. So, failure to recognize antigens of your own muscle cells. Okay. Now, as a result, what happens is it triggers an immune response, which would be the creation and secretion of antibodies to match that particular antigen. Okay, so then you would have antibodies, okay, are created that are specific, complementary. Okay, that's a good word to use, complementary. Some places require it as an actual word. Two antigens. Okay. What happens is that these antibodies then bind to these antigens. Okay, these antigens could be receptors or anything like that. And this therefore prevents any kind of, any chemical transmitter molecule. Okay, so example, neurotransmitters from binding. Okay, so I've actually run out of space here. So I'm going to say from binding. Okay, and if it cannot reach and bind, then it does not stimulate or trigger the response that it normally would. So you could say then here prevents desired response, because it's not actually being, the information is not being passed on to the cell because the antibody has blocked that transmitter molecule from getting to it. Okay. Okay, so you just really need to describe the induced fit hypothesis of enzymes here. The rest of the context is just to confuse you. So what happens here is that you could say that the active site, okay, you must state the active site of Y is not fully complementary. Okay, it's not fully complementary to the cell signaling molecules, to the cell signaling molecules. Okay, so what happens here is that once the substrate fits into the active site will change shape. So the active site molds, you could also say changes to fit the substrate. Yeah, so to successfully attach to the substrate, it will mold around the substrate to become complementary in that sense. Now, what happens there is that once this has happened, it forms an enzyme substrate complex. Okay. And then after the reaction has taken place, the products are released from the active site. Okay, and then the enzyme can keep going about its business. Okay, you can also mention things like lowers the activation energy here. You know, that's a main function of enzymes anyway, but here would guarantee you a full mark. So the first mark is for the active site not being fully complementary. The active site changes and fits the mold. The idea, once it happens, there forms an enzyme substrate complex. And then after that, the reaction, the products are released from the active site, and then the enzyme is unchanged and left about to go do its business. So in regards to this is referring specifically to the treatment of the disease. So it's clearly been identified that someone has the disease. You don't need to talk about how it's used to identify or anything like that. It's talking about the physical treatment, okay? Well, first of all, you should be aware that the monoclonal antibodies, the MABs, so first of all, they're not recognized as foreign antibodies, okay? So they're not recognized as foreign, okay? So they're not going to be destroyed by your immune response when they are injected into a person, okay? Now, these are, or should we say, these monoclonal antibodies are very specific, okay? So they're very specific to only the B lymphocytes that have the CD20 protein, okay? So they're very specific. They're not going to start attacking other cells or causing any other problems there, okay? So what happens is that these antibodies, okay, will bind to these, or to this protein, to these proteins, okay, the CD20. And what happens is that they can then be identified for destruction by phagocytosis and so forth to remove them from the circulatory system. This means that the corrupted, or should we say non-functional lymphocytes that are interrupting your cell-to-cell recognition can be destroyed and removed from the process, okay? So this can therefore lead to those B lymphocytes being destroyed by phagocytosis. Phagocytosis. Okay, here's 4 marking points. That's fine. This is more than enough for you to do. But just remember about 4 points. Always give yourself a little bit of extra room for an additional mark, just in case some of your wording might have caused a loss of a mark or been a bit too vague. Okay. Thank you for watching today. Please register on the website for a free demo, and I look forward to seeing you in our next video.